Look at this animated view of early Earth. It is a violent, desolate place. For centuries, people believed that complex life, like a rat or a maggot, could simply poof into existence from rotting debris. We now know that spontaneous generation is impossible. Instead of sudden magic, scientific focus shifted to a gradual, chemical process. Charles Darwin originally proposed that life might have started in what he called a warm little pond, where simple chemicals slowly combined under the right conditions. This photograph shows Russian biochemist Alexander Oparin. In the 1920s, he and British scientist J.B.S. Haldane independently expanded Darwin's idea into the modern chemical theory. They proposed that the early oceans acted as a vast primordial soup. Their hypothesis was that the origin of life was a slow, sequential cascade of chemical reactions. To make those specific reactions happen, early Earth needed an exact and very extreme set of environmental conditions. Here is an exam key breaking down those exact conditions. On the left panel, we have the physical environment. The prebiotic Earth was incredibly harsh. High heat from volcanic activity, constant lightning storms, intense UV radiation, and a strictly reducing atmosphere. That means there was absolutely no free oxygen. On the right side, we see the specific chemical inventory floating in those early oceans: water vapor, ammonia, methane, and hydrogen gas. When you trap those simple inorganic molecules inside an oxygen-free environment and blast them with massive amounts of thermal and electrical energy, the atoms are forced to break apart and forge entirely new chemical bonds. This flowchart maps the theoretical steps of chemical evolution, or abiogenesis. In stage 1, free atoms in the early atmosphere fuse together to form the simple inorganic molecules we just listed. Stage 2 occurs when extreme heat and lightning drive those inorganic molecules to react, combining them into simple organic molecules, or monomers, like amino acids and simple sugars. Moving to stage 3, those simple organic monomers link together to create long, complex organic polymers, such as proteins and nucleic acids. Finally, in stage 4, these complex polymers aggregate into clusters called coacervates, or protobionts. These act as the immediate, self-sustaining precursors to living biological cells. This specific chronological progression successfully bridges the gap between non-living chemistry and biological structure. However, for decades, the Oparin-Haldane model remained a purely theoretical hypothesis. It required a physical test. This photograph shows Stanley Miller running a laboratory apparatus in 1953. Working with Professor Harold Urey, Miller built a miniature early Earth in the lab to physically prove chemical evolution was possible. Looking at this vector schematic of their apparatus, we start at the bottom left. A flask of water is gently boiled, simulating the high temperatures of the ancient ocean. As the water evaporates, the vapor travels up through these connecting glass tubes and enters a much larger atmospheric chamber. This upper chamber is the critical test point. To accurately simulate the prebiotic atmosphere, they injected specific gases: methane, ammonia, and hydrogen. Crucially, these were mixed in an exact 2:2:1 ratio. To provide the necessary activation energy, they fired continuous electric sparks through the gas mixture, simulating the relentless lightning storms of early Earth. The newly formed chemicals then pass through a cooling condenser, simulating rain. This liquid washes down and collects in a U-trap at the bottom of the system, eventually turning into a murky red fluid. When Miller and Urey analyzed that red fluid a week later, they found organic molecules. By exposing those simple inorganic gases to energy, they successfully synthesized amino acids, the fundamental building blocks of protein. Now, pause the video and try an active recall sketch of the full Miller-Urey apparatus. Make sure you label the boiling ocean, the atmospheric chamber, the spark electrodes, and that crucial 2:2:1 gas ratio. On an exam, questions about the steps of chemical evolution require the 4-stage flowchart from free atoms to coacervates. For questions on the Miller-Urey experiment, structure your answer using 4 bold headings: apparatus, procedure, observation, and conclusion. Here is a complete cheat sheet summarizing the testable data points. It connects the Oparin-Haldane hypothesis to the Miller-Urey physical experiment, highlights the exact gas ratio, and identifies amino acids as the final result. Mastering this sequence gets you the points on your exam. It also demonstrates exactly how the raw, inorganic elements of a dead planet successfully combine to create the organic building blocks of life.